Hello and welcome to this video on how it is that RNA and DNA is read as such that it can be translated into proteins or regulate other bodily functions. DNA is the basics of who you are. It is the totality of every chromosome, gene, codon, and nucleotide. If you could just take any part of the DNA strand and turn it into protein, you wouldn't be able to regulate the size, and the size of DNA would be massive. This is why DNA is tightly packaged. That packaging makes it hard to read. In order to take DNA, or even RNA, and read it in a particular way, you need to do certain things, or more accurately, you must be able to do certain things. Otherwise, it would be like writing gibberish. It's why most languages have a set method of reading it. If you don't have that, there is no end result, and if there was, it would be entirely unpredictable. Using DNA as an example, the three nucleotides T, A, and G create a codon, or in other words, the three nucleotides needed to send a stop signal to whatever is being translated. If instead it was read as GAT, the opposite direction, it would produce histidine. If instead of stopping a gene being expressed, you just add histidine to the end of it, you might just create a random shape and the protein no longer functions as intended. That is just one amino acid, let alone what might happen for a whole intron, exon, gene, or regulatory function if you did not send a stop signal, let alone any other unpredictable outcomes. There is also a rather pragmatic issue that DNA is made up of two opposing sets of nucleotides. These match each other. If you read both strands of DNA in the same direction, you would get two different sets of products. This raises the first question of how DNA or RNA in your body is read, these both being certainly far more literate than some people anyway. The mechanism for this is called a reading frame. The frame reads in two directions based on which strand we are talking about. However, the DNA is always read from the 3' to the 5' direction in the first strand. This results in RNA being read in the reverse direction because of how it gets translated from DNA. This means to get coherent meaning out of RNA, and not view it as a toddler's scribble, it must be read in the 5' to 3' direction, remembering that you've made a copy of what is being made, so it's being mirrored. In order to understand it, you need to read it in the opposite direction. For DNA, we have two opposing strands of DNA, and the opposite direction is very similar to what you would get with RNA. That is, if you try reading one strand, the opposing nucleotides facing you on the other side need to be read in the opposite direction. In order to find the right place to start translating DNA or RNA, you need a start codon. This is another reason for sense in RNA and DNA. It prevents random start locations or random end locations, ensuring you get a complete read of the gene or whatever else it is you are trying to express from your bit of DNA. Now for the actual reading process of DNA, this is slightly more complicated than just directionality. First, in order to get DNA and if you want to transcribe it to create more DNA or translate it to create RNA of some kind, you need to unwind the region of the DNA we're talking about, remembering it's tightly bound up in that double helix and then wrapped around histones to create a chromosome. The region that you end up unwinding is called the transcription bubble, and this is the area where you're going to start reading. Depending on which side of the DNA strand we're talking about, this is where we're going to get the template strand from, and you'll be getting it from one of the two DNA strands. You rarely if ever copy both. The only time that ever really occurs is if you are synthesizing DNA for cell division, which is a unique scenario itself. Nonetheless, you take the DNA and you read it in the correct direction, this generally being the 3 to 5' direction. Once you have this DNA copied, you now have the primary template strand that is then used to match the opposing side, so you don't even need to copy the DNA itself from the original DNA strand. You just find matching pairs like a child's game of Snap. The only difference between this process for creating more DNA and for creating RNA is that RNA contains a uracil nucleotide rather than a thymine nucleotide. Thymine is found in DNA, uracil is found in RNA. If we're talking purely about taking DNA and turning it into RNA, this is done via an RNA polymerase that goes down the DNA template strand, and that's how it creates RNA, going from the direction that it needs to travel from and to. Once the gene or whatever else is translated, transcribed, or otherwise copied as needed, polymerases go back through and tighten everything back up and disassociate the various enzymes from the DNA template, and if need be, from the RNA itself. In order to get the enzyme to leave the strand, they generally use something like a series of repeating nucleotides that cause the enzyme to stall and therefore leave from the strand of nucleotides. RNA is slightly different, arguably comparably easier in terms of how it's read, and this again is done in a direction, to be clear about it, and for exactly the same reasons that you don't want DNA being read incorrectly. However, with RNA, the slightly smaller difference is that it goes through a ribosome, and the ribosome is essentially an enzyme whose sole job is to take the various amino acids and put them together under the instruction of the RNA. These RNA units are called an anticodon. The anticodon is basically telling the ribosome, here's the amino acid, now find it and plug it in, and we can create a polymer or long series of these amino acids. They are passing through the ribosome in a predictable manner, and because of how they pass through the ribosome itself, that process leads to the hydrolysis of the GTP bound to the ribosome itself. When that energy is processed via the hydrolysis, it forces the ribosome to effectively turn, and that turning process pushes the codons forwards. At the end of those codons, you get the next one coming up, and so it's almost like belt feeding for a machine gun, except rather than making bullets, you make protein. Because you move them down one at a time, you always get the right codon in the right place. If, however, and this is the big if as far as sense goes, you are instead to read it in the reverse direction for whatever reason, you may inadvertently get an entirely different kind of protein, or more accurately amino acid, being put in that place. And if you get a series of amino acids in the wrong place, and they're relatively close to each other, it is entirely possible that what you end up creating will not form the correct way for that protein. This is one reason why the sense is so important, other than what we mentioned earlier about things like stop signals. But this is not just the only issue, and it's why DNA and RNA need to be read the right way. And by reading in one direction on one strand of DNA or RNA, and the opposite direction on the other strand, you always get the right product for what you're trying to achieve. It also means that genes and similar always function as intended. But once you start getting out-of-place items, specifically nucleotides, although it can be much larger sections of DNA, that gene no longer performs its job, and that can open the door to things like cancer, misfolding of your cells, like for example a sickle cell anemia. All these issues because of a misreading of the DNA. And so this is why directionality of reading for a DNA or RNA strand is so important. Without that, it would be like reading a sentence, but not knowing whether you meant to read the sentence left to right to left, up to down, or down to up. It's simply impossible to get any sense out of what you're seeing. Thank you for watching this video. If you found it interesting, consider liking, sharing, and subscribing. Please do post any comments, questions, or suggestions you have below.